Hey guys, Alex here, and I'm going to talk about maintaining a colony of Spodoptera exigua for feeder insect purposes. Um, just a quick thing, when I did the actual show of the colony that we're maintaining, the sound didn't record, so when I show that part, it's going to be me narrating over the original video, so it might be a little clunky, but it should give you the idea of what we're doing. So, Spodoptera exigua is a moth in the Noctuidae. It's about twice the length of a blue bottlefly, and it is a fairly easy moth to rear. It functions mostly like a nododontid, and the reason I say that is because nododontids don't feed as adults. This guy doesn't need to be fed as an adult, even though they will eat as adults. They will still breed without feeding as adults. The adults are great food for mantises, especially larger mantises and things that really like to eat flying insects. I use the eggs as food for ladybugs, and we use the larvae as food for our assassin bugs. Spine soldier bugs will eat the larvae and pupae. My axolotls will eat the pupae. My frogs will eat the larvae. It's all around a good feeder insect. Spodoptera exigua is native to the Southeast Asia. It is an invasive greenhouse and crop pest in many parts of the United States. And it's highly polyphagous, which makes it really easy to keep and take care of. And we're going to talk about a couple different things we can rear them on and what we do here. So, Spodoptera exigua life cycle starts with the egg. The egg incubates for between 2 and 4 days, sometimes 5 days, depending on the temperature. We usually have ours hatch in 3 or 4 days. We usually rear them in the upper 70s Fahrenheit. If we're at higher temperatures, development will be quicker, and if we're at lower temperatures, development will be slower. What we should caution you guys about is rearing them at too cold of a temperature if you're using artificial diet, because artificial diet will kind of get moldy and bacteria laden at lower temperatures more quickly. For some reason, it just doesn't work out as well for them at lower temperatures. More on that in a few. The egg hatches into a larva. The larvae start off as a very clear, blandly colored, tiny caterpillar. They pass through 5 instars like most lepidopterans. They start to develop color in their 3rd instar. Sometimes they're green, sometimes they're yellowy, sometimes they're brown. They might have stripes, they may not. They can be a bit variable. The larval stage lasts anywhere from 7 to 15 days, depending on the temperature. Ours typically take about 9 or 10 days to reach full maturity, although sometimes we, even at higher temperatures, will get a portion of our larvae to become fully grown after just, like 8 or 9 days, and then there will be stragglers that pupate after like 14 days. We then have the pupal stage that lasts about 7 days regardless of temperature. The adults then emerge. They, within a few days, mate and they start laying eggs. The adults live about a week, with or without food. The females will lay egg masses containing anywhere from 10 to 300 eggs. Sometimes they coat the eggs with their scales and body hair, sometimes they don't. The egg masses are a grayish color. They turn dark right before hatching. And so, rearing them. What we do is we take a large plastic bin that's airtight because they're escape artists, and we line the bottom of the large bin with paper toweling, several layers of paper toweling, and then we put a slightly smaller bin that has the artificial diet at the bottom in there before we add the eggs or the hatchlings. Today we added some hatchlings because we ran out of diet. We had to wait a couple days for new diet to come in, and some eggs had hatched. That was fun, but we got our work done here. And we then put a lid on, and we have, a large bin that maintains a humidity space. The condensation that is released from the diet will go and fall back down into the paper toweling. Every few days we'll change out that paper toweling as it gets wet. Humidity is okay, but standing water causes a lot of problems. And so that's why we like to keep the paper towels at the bottom dry, but we keep the humid space around the larvae so they develop well. The larvae will then burrow into the bottom of the paper towels or into the bottom of the artificial diet, where they will pupate. We will harvest the pupae once basically all the larvae have become prepupae. So what you'll see is we have some larvae that are prepupal, some larvae that have pupated already. There is some difference in their development rate. It happens. They don't always hatch at the same time. What we do is we will go and harvest them. We then set them up in bags to emerge and mate. We take the bags and we put about 20 pupae per little portion cup that has a moist piece of cotton on the bottom of it. We take that, we put it in a bag, we put a portion cup with a very wet piece of cotton next to it in the bag, and we close the bag up. About a week to 8 days later, the adults start emerging. About 3 or 4 days after that, they'll be mating and laying eggs. Once they start doing that, we move them to a new bag that doesn't have the cups at the bottom anymore. They'd like to lay their eggs in the cups, which is annoying, and we then will keep our rotation going. We like to stagger our hatching, so what we'll often do is we will take a bag and we'll put adults in it. We'll collect those eggs a day later, and then the next batch of eggs that come out from the adults the next day, we will freeze or something, and then we'll put the adults in a new bag. And then those eggs that are a couple days older than the first eggs, a couple days younger, will be used. So we'll have bins of eggs that hatched, maybe on Monday, and we'll have a bin of eggs that hatched on like a Thursday, and then a bin of eggs that hatched the following Sunday, so that at all times we have larvae of different sizes, different ages, and that really helps us have a constant flow of larvae that are appropriate size for our insects. If you want to rear them on foliage, that's fine too. There are a number of different plants that you can feed them: cabbage, spinach, kale, collard greens, probably lettuce, and the husks from corn you can use. Make sure you're getting something that's organic because you don't want pesticides. What I strongly, strongly recommend when you're rearing any Lepidopteran on host leaves is to get a spray bottle with 2% bleach. 2% is all you need. Spritz the, mist the leaves with that 2% bleach. Shake it dry quickly, and then throw it in there with them. What I would do is have a bin where you have a couple layers of paper towels on the bottom. You put the leaves on, you put the larvae on the leaves, and as those leaves start to go bad, remember that this is herbaceous plant material, so it's going to release a lot of moisture. What you're going to want to do every, couple days, depending on how quickly they're eating the leaves and how quickly the leaves are getting ratty, is put a new layer of fresh, clean, healthy leaves on top of the larvae. Wait for them to start moving onto that. Then pick all the new stuff up, set it aside with the larvae on it. Throw away the old paper toweling, the old foliage, wipe it down, and then put some new paper toweling down so that you're catching excess moisture, you're keeping things nice and clean, and that will prevent a lot of problems. What we do is we have the adults in bins, in bags. We don't feed them. We don't do much more than give them a spritz of water whenever we're moving them around. Every couple of days in the pupal bag, we add some water with a spray bottle, and they're fairly low maintenance. I should quickly clarify that when I show you the colony setup, the larvae from a bin that I'm harvesting, the prepupae and the pupae, that particular bin had a bacterial outbreak. The diet was a weird color. I think it may have been too moist in there for them. We did get some prepupae and pupae without a problem, but we also lost a lot. That happens every couple of generations. But what I will talk about now are commonly encountered problems, and there are a few. So the first, obviously, disease outbreaks. So there are many different artificial media available for people to use, and most of them contain some sort of bacterial or microbial inhibitor, antibiotics, things like that. Make sure that when you get any of these artificial diets, you get one that has some sort of inhibitor in it so that you can prevent as many different diseases as you possibly can. You obviously want to keep humidity levels reasonable. So again, preventing infections includes things like making sure that when the paper towels at the bottom are drenched, you change them out. You prevent standing water and condensation from being a problem in the bins. You use diet that has an inhibitor, or if you're using foliage, you're spritzing it with some bleach before you feed it to them. Don't worry, it won't harm them if it's 2% bleach. we do it with all of our caterpillars. Whenever I were basically anything, I spritz that foliage down with some bleach. Then you want to kind of keep things clean. Bleach is your friend. I make sure that when I am done with the generation, I either dunk or spray down all the materials, all the cups, all of the bins and everything with 10% bleach. Make sure that 10% bleach has contact with that surface for at least 10 minutes. It does help to kind of rinse and scoop any kind of organic material away before you spray the bleach down so that it's as bare as possible. Put that bleach on it, let it soak, let it sit, and that should really keep pathogens at a minimum. Another problem that we encounter a lot is the first larvae, the pupae, sometimes get cannibalized when they're pupae by the other larvae. Even if there's plenty of food around, for some reason, larvae really like to eat the pupae. And this isn't a big deal for me because I have a bin that has, 250 plus mature caterpillars. A few are going to pupate early, and they might get munched by some of the other caterpillars. But after a few days, they're all basically prepupal, and so you're really not going to have too much loss due to this. If it's really that important, removing the pupae as they form does reduce this from happening. Like if I'm doing an experiment with them, that's definitely what I'm going to do. But if I'm just rearing them out because I'm using them as feeders, we don't have such a huge loss from this that it's going to cause problems. A lot of this is a numbers game. We sometimes lose a good number of our larvae, like this past bin that you're going to see, about 2/3 of the larvae died of an infection. But we still got, several dozen pupae and prepupae, and that's more than enough to start another generation. It's not the only bin that we took pupae from, because we have multiple bins going, and, we end up producing so many extra pupae and things like that. We end up feeding off or killing extra pupae. We end up freezing extra eggs, even if we start with a small number of pupae that emerge and we allow to mate. They're highly prolific animals. And so, I haven't yet had a disease wipe out the entire bin of larvae. I've had, some disease problems every few generations, but it generally doesn't cause too many problems. Another problem that we sometimes have is phorid flies laying their eggs in the media. They're not that harmful, but they do kind of make a mess of the media. And so what we generally do if we notice phorid flies is after we've collected the pupae and prepupae is we just take the diet and we freeze it. And that generally kills off the phorids and that suppresses them. Kind of just excluding them to begin with is the best way to do that. And so there's that. Those are the biggest issues that we have. Sometimes pupae don't emerge. That has more to do with the temperature, the humidity that they're experiencing, the bags. So yeah, we basically have artificial diet that we make. We buy the powder for it. You can generally make artificial diet quite easily. If you really want to, what you should do is get the artificial diet, add the proper amounts of water and the diet powder, and then microwave it until it's really boiling. Use an oversized container so it doesn't boil over. But what I would do is microwave it rather than use a stovetop or a burner or anything, because the microwave will help sterilize that inside of the medium, and so the artificial diet will be a little bit cleaner, last a little bit longer, and you'll get it nice and boiled so that all the gelling agent dissolves, everything is mixed thoroughly and activated, and you're ready to go. There are a number of different commercial artificial diets available for rearing Spodoptera. These are really generalists, so it's not that difficult to find an artificial diet that they'll eat. We've used a couple different types, and so that's about it. We make the diet, we let it cool and harden, we put the eggs on it, we allow the larvae to develop, we pull them out to feed them to things as needed, we have them then, we allow some to mature, we harvest the pupae, we put them in little cups with some cotton that's moist, we put them in paper bags, we allow the adults to emerge out and mate, we cut up the bags to collect the eggs, and the cycle repeats. So now you'll see kind of our colony action, and I hope this helps people. So as you can see here, we have a large bin, and inside of that large bin are paper towels lining the bottom, and a smaller bin that has artificial diet. The artificial diet was made earlier today, and now it's cooled and hardened, And now we can add the eggs / neonate larvae. And so I'm going to do that. Paper toweling on the bottom to collect condensation. The lid doesn't have any holes in it. If the lid does have holes, I cover it with plastic wrap. Here we go. That's a cup that the adults laid their eggs on. It's annoying when they do that, but they do that. More eggs. See the egg masses along the side of the cup. More. And then I have a whole bunch of pieces of paper bag that have the egg masses. As you can see, some of them have the scales from the mothers, some of them don't. Some of those bigger egg masses probably contain a few hundred eggs. The eggs are fairly small. I try to put as little paper bag material in there as possible because it molds very easily. So I'm going to then cover the container with the lid. Make sure it's completely on tight. Then I'm going to label the bin. Use the labeling tape. Then I write the species and the date, just so that I can keep track. There we go. So now it's time to put the bin up on a shelf. No space there. Here I go. We're good. Alright, so now we're going to harvest some prepupae and pupae. Here we have an older bin. This bin had some bacterial issues, as I said before, but we're just going to roll with it. I'm going to pick up the bin that's inside the larger bin, get anything off the bottom of that inner bin. I'll harvest the pupae and prepupae. What I need first are some small portion cups. These are 2-ounce portion cups. I'm going to need some cotton padding, some distilled water, some scissors, and we're going to get going. The cotton pads are fairly large, so I cut these ones into fourths. We also have smaller cotton pads that I cut in half. I'm going to set up the cups 2x2. Do-do-do-do-do-do-do. Cotton pads. We're going to want to cut these into fourths, see how big they are. Then we're going to want to put them in the cups. Then I'm going to take my water. So half of them I'm going to make slightly moist, just enough to have moisture on them, and then the other half I'm going to make very wet. Notice how they're paired. You'll see why this is the case in a minute. So we have moist and wet cotton. And then I'm going to add the pupae. I'm also going to add prepupae to half of these because I want to stagger my emergences. The prepupae are going to take another day or two to actually pupate, so they'll take a couple extra days to emerge into adults, and thus they'll produce eggs a little bit later. So I'll add 20 pupae to each of the cups and 20 prepupae to the other cups. So each moist cotton cup has either 20 pupae or 20 prepupae. That's plenty. Here I'm adding prepupae from the diet. It shouldn't be that color. It probably was just too humid in that bin. Sometimes this happens. I still got plenty of pupae to harvest. Now here we're going to take a sieve. I have a sieve that has very small holes. I put a sieve that has very large holes. And then I take another sieve with large holes and overlap. I'm going to pour the contents of the diet bin into the sieve. And I'm going to shake it out over a garbage can. You can see the prepupae and pupae falling into the sieves. And this is just going to separate the pupae and prepupae from all the waste material. It's fairly moist, so it doesn't separate as easily, but you get the point. Alright, so now I'm going to take my paper bags and some binder clips. And what we're going to do is we're going to take a bag, we're going to open it, and then we're going to take one moist cotton cup that has the pupae or the prepupae and one wet cotton cup and put it next to one another in the bag. We're going to close off the bag and seal it shut. Well, not really seal, but close it shut with the with the binder clip. And we're going to keep doing that until we have all bags set up. Alright, so now I'm going to show you the adults. Here's a bag where the adults have emerged, and I've moved them into a new bag. So you can see the adults in there. That's about the size they get. You can see an egg mass right there. So they're reproducing quite well. I'm going to give these guys a spritz of water just to keep them hydrated. They don't need to be fed as adults, but having a little bit of water does help them. And I'm going to do that with all the bags that have adults in them. That's the shelf where I keep them up on. I'm going to show you the neonates next. I'm going to put my bags away. Alright, so this is a slightly younger bin. I set it up on the 24th. On Saturday, which was the 26th, the larvae started hatching. You can see them all covering those paper bags. In a day or two, I'm going to pull all that old extra paper bag material out, but you can see all the very small young larvae scattered across the diet and the bin. I'm definitely going to want to remove those paper bags in a day or two because they will really mold over and cause problems.